Hello everyone and welcome back. We are going to cover part 1 of chapter 4 today, the functional and anatomy of prokaryotic and eukaryotic cells. Please keep in mind that a majority of this chapter is going to be focused on prokaryotes because you've learned a whole lot about eukaryotic cells in your discussions in intrabiology as well as in anatomy and physiology. So we won't really spend a whole lot of time talking about eukaryotes, but we will spend a wealth of this chapter spent discussing the functional aspects in the anatomy of prokaryotic cells. So let's do a little bit of compare and contrast. So for prokaryotic cells, they are much simpler than eukaryotic cells, and their genetic composition really just consists of one circular chromosome, which is not in a membrane-bound organelle, which is a little bit different from what we find in eukaryotes, that they actually have a membrane-bound nucleus that holds the genetic material. And when we say genetic material, remember we're talking about DNA. Prokaryotes don't really need histones, which are those special proteins that are responsible for making sure that the DNA stays nice and tightly wound and coiled up. Because the DNA is not that complex, there really isn't a need for histones. However, there is a need for histones in eukaryotic cells. Eukaryotic cells do have histones. Prokaryotic cells have no membrane-bound organelles, so no Golgi apparatus, no lysosomes, none of those structures. Eukaryotic cells do have organelles, lysosomes, Golgi apparatus, etc. Bacterial cells or prokaryotic cells, if it's a bacteria, then it has cell walls that are made out of peptidoglycan. And we talked about in our very first chapter how not only can bacteria have cell wall, but we can also have eukaryotic cells, such as plants that have cell walls, and fungi can also have cell walls. Now, for eukaryotic cells, their cell walls aren't made out of this protein sugar complex. That's what peptidoglycan is. Their cell walls are made out of a polysaccharide. And that polysaccharide can be chitin if we're talking about a fungus, or it could be cellulose if we're talking about a plant cell wall. So we're just going to generally say polysaccharide cell wall. Prokaryotic cells can also be archaea. So remember, not only are bacteria prokaryotic cells, but so are archaea. They have cell walls as well, but their cell walls are made out of a different material called pseudomurin. And the way that bacterial cells are going to divide is via binary fission, whereas eukaryotic cells use a mitotic spindle. So here we have the basic shapes of bacteria, and I'm not going to really read much over this. I'm going to allow you all to read this because we looked at this in lab and we talked about this on our very first lecture. So if you don't remember this, please refer back to the Chapter 1 lecture where we discussed this at length. And once again, I won't spend much time talking about this, but I'll just show you the slides that we can have different arrangement of cocci. They can be two together in pairs, or they can be in chains. If it is in chains, we call it strepped. If it's in clusters, then we call it staph. Same as with bacillus, we can have a single bacillus or rod shape, or we can have them a coxal bacillus, which has both a coccus aspect to it, and it's also long and rod-like as a bacillus would be. And then we have our third shape of our traditional shapes of bacteria. The vibros, cholera is a vibro. We'll talk about that in one of our final chapters. It kind of has a curved shape to it. We have the sporulum and the spirochetes. Syphilis are great examples of spirochetes. Having that kind of cork shape configuration allows it to corkscrew its way through very viscous materials such as seminal fluid and vaginal fluid, which makes it an excellent sexually transmitted disease because it's able to get past some of those barriers that you have in your sexually reproductive organs. So bacillus is rod-shaped. The arrangements could be two cocci together or two bacilli together, hence the name diplobacilli and diplococcus. Clusters, this is so important. I'm going to star it because you're going to talk about this kind of throughout your microbiology career. Clusters are always known as staph flow something or another. So you can have Staphylococcus or you can have Staphylococci, and chains are considered strept. So Streptococci would be chains of balls or chains of spheres together, and Streptobacilli would be chains of rods. And we'll get a chance to look at that in lab once we do some gram staining. So remember, staph, cluster, chain, strep. So what we're going to do now, before we dive into the other structures or the structures that you would typically find in a prokaryotic cell, is that we're kind of going to look at this roadmap. So anything that is labeled in red are going to be structures that you find in all bacteria. The other structures that are labeled in black, we'll still discuss them, but keep in mind that not all bacteria have a pilus or have a flagella or have a cell wall or have a capsule or inclusions or any of those that are listed in black. So I'm going to give you all the opportunity to just look over these structures, because I'm sure it's not the first time that you've seen it. And then on the next slides that we look at, we will talk about the purpose and the role of each of these structures. All right, so we're going to work our way from the outside in. And on the outside of some bacterial cells, you have what's called a glycocalyx. The glycocalyx is always going to be found outside the cell wall and has a very sticky aspect to it. There are two jobs that a glycocalyx can have. It can help the bacteria to adhere to other bacteria, especially important in a biofilm, or it can help the bacteria to adhere to surfaces. So on the insides of pipes, on your teeth before you brush them, on the inside of your toilet in your shower, you have a biofilm, and it is the glycocalyx that allows for these bacteria to stick to these areas. When we refer to a glycocalyx, it's just a very general term that can either be represented as a capsule, which is very tightly organized. Capsule's main job is to prevent phagocytosis from taking place, so it makes the bacteria much slipperier, so that when you have your macrophages and other phagocytotic cells that are attempting to eat that bacteria, it can't really get a hold of it. So that's really the job of a capsule, and we'll be doing a capsule stain in one of our labs coming up real soon here. We also have the type of glycocalyx that can be a slime layer. Slime layers are very unorganized and very loose, and as opposed to capsules, which make it difficult for phagocytosis to take place, a slime layer is really mostly configured to allow it to stick to surfaces. The extracellular polysaccharides allow the cells to attach to either one another, or they allow the cells to attach to surfaces, specifically in the case of a slime layer. And as we said before, the capsules prevent phagocytosis. So here we have Streptococcus pneumoniae, and notice that it has a capsule around it, and it would be considered a diplococci because it's paired cocci that we have there, and this is our capsule that helps it to evade phagocytosis. So when you have this bacteria gets into that what you would like to be sterile environment of your lungs, there is sometimes it's able to surpass that or get past that because it has that capsule layer. Next structure we'll talk about is the flagella. Now keep in mind, remember when we looked at that first page where we had the different structures on a prokaryotic cell, that not all prokaryotic cells, not all bacteria have a flagella. But if you are fortunate enough as a bacteria to have a flagella, it's going to be attached to the outside wall. It's made out of special chains of a protein called flagellin, and it attaches to a protein hook in a couple of different arrangements, depending on whether the cell is gram-positive or gram-negative. So it can be anchored to the wall by what's called a basal body. How thick that basal body is or how complex that basal body is depends on whether the cell is gram-positive or gram-negative. Typically, gram-negative cells that have a flagella have a much larger or more complex basal body because there are 2 points of attachment to the plasma membrane and also to the cell wall, as opposed to gram-positive cells, which have a less complex basal body. So here we have the attachment of a flagella on a gram-positive cell. So that basal body that we're talking about is that point of attachment that happens here. So for cells that have a cell wall, for bacteria cells that have a cell wall, they're going to have a plasma membrane, and then they're going to have a cell wall configuration to them. So this is a gram-positive cell. It has a very thick cell wall, but it only has the cell wall. It's like in the beginning of that bacterial cell. So the basal body that we have here is actually quite simple. And when we compare that to a gram-negative, you're going to see a little bit of a difference. And I'll talk to you and tell you why that is in just a sec. So now when we look at the gram-negative bacteria, notice that just as a gram-positive, they have the phospholipid bilayer, also known as the plasma membrane, but then they have their cell wall, which is illustrated here in brown, and then on top of the cell wall, they have this outer membrane. That's what we call the lipopolysaccharide layer. Because it has this additional membrane, the basal body is going to need 2 points of attachment. A point of attachment at the plasma membrane, where we saw for the gram-positive, but it's also going to need a point of attachment in between the cell wall and that outer LPS layer. It's this outer LPS layer that's the reason that gram-negatives are much more difficult to kill with soaps, detergents, and just run-of-the-mill antibiotics. And it's also the reason that when you gram stain them, the outer layer washes off with alcohol, and we counter stain them with the saffron. So the way that the flagella can be arranged in prokaryotic cells is very different than what we see for eukaryotic cells. So for eukaryotic cells, there's really only one arrangement of the flagella, and it's considered monotrichous and polar. People, or those folks that have flagellated cells, are only males. So their flagella just looks like this. However, for bacteria, it can be peritrichous, peri meaning all around, trichous kind of means hair-like, it's Latin for hair. So you have these peritrichous, which means the flagella is all the way around. You have a lophotrichous and polar, which means it's a tuft of flagella at one end, so more than one flagella, but it's only at one end, and there are many flagella that are there. Or you can have amphitrichous and polar. If you're ambidextrous, that means that you can use your right hand and your left hand, or your right leg and your left leg equally. And then polar, it means that they're found on both ends. So the way that bacteria would use their flagella is in what we consider run and tumbles. Unlike eukaryotic cells, where their flagella, and I'm going to attempt to draw this for you, eukaryotic cells, their flagella moves in a whip-like fashion. So they're going to move in this kind of up-down sort of whip-like fashion for eukaryotic flagella. For prokaryotic flagella, they kind of move more in a rotation. So they rotate around in circles, and that's what facilitates the movement of eukaryotic flagella. They exhibit what we call positive and negative chemotaxis, which means that the flagella will propel the cell either towards a stimulus that they want, food, for example, that would be positive chemotaxis, and away from a stimuli that is negative. So in the case of antibiotics, that would be a negative stimuli, so it would exhibit negative chemotaxis, or to run away from it. And they do so not just in a straight line, but in a series of runs and tumbles. So when they go away in a series of runs and tumbles, they'll kind of go towards it, and then they'll turn around in a circle, and then they'll go, so if this is where we're trying to get to, that's the money spot, then they'll kind of make this direction, and then they'll have a tumble here, and then maybe go in that direction and have a tumble here, and then finally make it to where they want to be. Another important aspect about flagellas is that they help to give us antigenic determination, which means that the flagella on different strains of bacteria can have a special antigen or protein on it that is different from other bacteria. For example, the E. coli in your gut does not have this antigenic protein called H7 that we would find in the more pathogenic form of E. coli 15H7. So they have a role in antigenic determination. That's really what this is talking about. And when we say antigenic determination, that just means it has a role in specifying or differentiating one type of bacteria from another, even though they are the same genus and species. So here's a pretty picture of that run and tumble, and I won't have any discussion over this. I'll just let you look at this and kind of digest this yourself. this would clearly be an example of a peritrixis arrangement of the flagella. So flagella don't have to just be peritrixis, lophotrixis, monotrixis, and the other arrangements that we saw. We can have special types of flagella specifically found in spirochetes that are called axial filaments, or also known as endoflagellas. Instead of being anchored at just on the outside of the cell, they're actually embedded into the periplasma space between the plasma membrane and the cell wall. So although they are anchored at one end of the cell, they kind of go through that periplasmic space, and instead of just causing this rotation to happen at one end of the cell, the rotation is going to happen throughout the cell, and it causes the cell to move in a corkscrew type fashion. So here we have a picture of Leptospira, which is a spirochete, and you have the endoflagella that are embedded within that periplasmic space, and it will allow its way to kind of corkscrew itself through its media, in much the same way that syphilis is also a spirochete, and it also has endoflagella or axial filaments that will allow it to move through very thick, viscous material, as we'd find in semen and we'd find in vaginal fluid, which makes it conducive to cause a sexually transmitted disease. Other points of attachment, and before I go any further, I want you all to take note that we've talked about the Glycocalyx, capsules, slime layers, flagella, endoflagella, different arrangements of flagella, and we still have yet to get on the inside of the cell. So just a point I wanted to make so that you all realize how complex the outside of a prokaryotic cell is, relatively speaking, compared to the inside of it. The reason for that is because the inside doesn't really have a whole lot going on, and that prokaryotic cell's life and ends begins at its outer membrane. So it needs to have lots of structures that are meant to protect anything from getting onto the inside of that cell. Because if something gets on the inside of that cell and causes what we call lysis to take place, then game over. So other structures that we can find on a prokaryotic cell are Fimbriae and Pili. Fimbriae, like the Glycocalyx, may allow for attachment to either surfaces or other cells themselves, or the bacteria cells, especially important in the formation of a biofilm. And here we have some Fimbriae. They look more like sticks, whereas the flagella look more like hairs, so it looks more stickish in appearance. And it's much longer than what we'd see for the cilia that we see in eukaryotic cells. The Pili's job is meant to transfer genetic material from one cell to another. We could also call it the sex pili. So bacteria cells, even though they divide by binary fission, which is just basically mitosis, one cell becomes two, they should at that point be genetically identical. But we understand that bacteria cells are not all genetically identical. If they were, we would have gotten rid of them all a long time ago. But they have some genetic differences between them. And one of the way that bacteria are able to have what we consider a horizontal gene transfer is through this sex pilus, where it's a hollow tube that allows for genetic material to be transferred from bacteria A to bacteria B. And I guess the way to think about the pili is that the sex pilus, through this process of conjugation, is as close to sex as bacteria are going to actually ever get. And we'll talk more about this in Chapter 8 in our discussion on genetics. Finally, after much to do, we have made it to the cell wall. The job of the cell wall, and I'm going to start this because once again I feel as though it's important, the job of the cell wall is to prevent osmotic lysis. Well, what does osmotic lysis mean? Osmotic lysis means the cell gets too much water in, so it bursts, explodes, and dies. Now for you, if one of your cells bursts, explodes, and dies, it's not really a big deal, even if it's a brain cell or a heart cell or a lung cell, because you got a whole bunch of other cells that help to make up that organ. For a bacteria, its life ends and begins at its cell wall or its outer membrane. So if it were to explode and die, then lights out, game over, shows up for that bacteria cell. And as we've already stated, the components that make up the bacterial cell wall are a combination of proteins, peptides, and sugars, glycan, that we call peptidoglycan. So here we have those structures that we've talked about, the capsules, the cell wall, the fimbriae, the pilus on the outside there. So really it's just kind of looking at that picture again, and I won't go over and explain all these structures because most of which we've talked about. Now the plasma membrane, cytoplasm, ribosomes, and the nucleoid, these are structures that are found in all prokaryotic cells. So here's a picture of what that plasma membrane, I'm sorry, not plasma membrane, but cell wall is made out of. That combination of proteins and sugars is made out of chemicals called N-acetylglucosamine, important, known as NAG. You'll be asked this on your exam, or I would say list the two components of the cell wall. You should know that one of them is N-acetylglucosamine, and then the other one, NAM, is N-acetylmuric acid. Not important that you remember the structural configuration of this, but you should know that N-acetylglucosamine and N-acetylmuric acid are the components of the cell wall. So let's look at some differences between a gram-positive and a gram-negative cell wall. Gram-positive cell walls and everything that I star or sunlight is really important. Gram-positive cell walls have a very thick peptidoglycan layer. Gram-negatives, they don't have as thick of a peptidoglycan layer. However, they're able to make up for that thin peptidoglycan layer because they have an additional layer to it, the LPS or outer membrane layer. Because of that, they're going to have much more periplasmic space. We do find that we have periplasmic space in gram-positive cell walls, but it's not as thick and it's definitely not as abundant. In gram-positive cell walls, we also have these anchoring structures that we call teichoic acids. So when we look at this gram-positive cell wall, remember one of the hallmarks of it is it has a very thick peptidoglycan layer. That peptidoglycan layer for both the gram-positives and the gram-negatives is illustrated as the brown layer, and the plasma membrane layer, or the layer that has phospholipids. Phospholipids are made out of, as the name suggests, lipids, so they're fats. As the yellow layer. If we were to look up close at this gram-positive cell wall, we find that we have the plasma membrane, we have the cell wall, which is made out of that NAM and NAG, N-acetylglucosamine and N-acetylmuric acid. And then we also have these teichoic acids. We have these wall teichoic acids that help to stabilize the peptidoglycan layer. And then you also have the lipoteichoic acids that will anchor the cell wall to the plasma membrane. When we look at the gram-negative, it's a little bit more complex. We have our plasma membrane layer in the same way we have with our gram-positive. Then we have our cell wall, and now we have this outer lipopolysaccharide layer, or an LPS layer. What are components of the LPS layer? Primarily, it's lipids and sugars, liposaccharides. You have core polysaccharides, O polysaccharides, which, like the H antigen, can be used for antigen determination or to determine one strain of a bacteria from another. And lipid A can act as an exotoxin. That can be released from the cell. And then you have this, majority of it is a phospholipid layer. Because it is a fat, alcohols wash fats away. So alcohols will completely dissolve the fat. So in our gram staining procedure, when we're doing our decolorizing, we use this acetone alcohol that helps to wash away this layer. So when we first gram stain, we noticed that both the gram positives and the gram negatives were purple. But then once we put on that LPS layer, then it got rid, once we put on that alcohol, the alcohol completely dissolved this LPS layer, so there's no more color left. That purple color gets washed off. So then we have to come back in, and now we can finally get to the cell wall, and we stain it with saffron. Because there is this outer layer, and you have this lipid A, it does protect the cell from detergents, soaps, and normal antibiotics, which is why for a clinical aspect, gram-negatives are much more resistant to treatment than gram-positives are, and that's a very important reason for gram staining. So the gram-positives have those teichoic acids, wall teichoic acids, link or attach the cell wall to the that keep the cell wall together, the peptidoglycan layer together. Lipotecoic acids are going to link the cell wall or attach it to the plasma membrane. We think those tecoic acids may help with the movement of positively and negatively charged ions that we call cations, things like sodium and potassium. And then the polysaccharides do provide some antigenic variation. So those tecoic acids that are found there can also provide some antigen determination in much the same way that we saw that we can have that from the O polysaccharide on a gram-negative. Gram-negatives have that LPS layer. Lipopolysaccharides consist of lipoproteins and phospholipids. What's one term that you see in each one of these words? Lipo for lipids. Lipids are fats. Alcohols dissolve fats. Make sure you remember that. There is a little bit of space between the that we call periplasmic space between the outer membrane and the plasma membrane. The LPS layer protects from phagocytosis, protects from complement, which are a series of proteins that are meant to either poke holes in the bacteria and make it explode, or to make it sticky for phagocytosis, or to cause inflammation. Your body has these complement proteins, so it's your body's defense against bacteria. Well, gram-negatives, because they have that outer membrane, they have a defense against that particular complement system, and also helps to protect it against antibiotics. And as we've already stated, the O polysaccharide helps for antigenic determination. So we'll go back to E. coli. H7 represents the special antigen on the flagella. O157 is the special O polysaccharide that's associated with this LPS layer, or outer membrane layer of the gram-negative cell. Lipid A is an endotoxin, which means that if you ingest a bacteria that has the lipid A as an endotoxin, then when your cell ingests that bacteria, the bacteria releases that toxin and it causes the cell to die. And then on the outside of that plasma membrane, or that in that LPS layer, you have special channels that will allow items to get through there. So even though we want to protect the cell from the outside world, there are still things like cations, sodium, potassium, glucose, things that are needed for nourishment that need to get onto the inside of the cell. So for the gram staining mechanism, I've kind of been talking about this. We spent a good chunk of Chapter 3 talking about this, and I've talked about it a little bit already when we talked about the differences between gram-positive and gram-negative cells. So here's kind of in words the process of gram staining and what each one of those stains and that mechanism does and how it actually works. So I'm going to let you all read this slide, and then I'll come back in because we've kind of been talking about it all along and give a little bit more detail. So for gram-positives, understand that the alcohol dehydrates that peptidoglycan layer, and what the iodine or that mordant does is it ensures that the crystal violet dye does not leave from a gram-positive. So that's why no matter what you do to it, no matter how much alcohol you put onto it, for the most part, unless you just completely drown the thing in alcohol, that the gram-positives are going to stay purple. For the gram-negative, the alcohol is not going to get to the peptidoglycan layer yet. It's only going to get to that alcohol layer. The same thing with the crystal violet iodine complex is really only staying that LPS layer. Because it's made out of lipids, and the just chemical nature and physics of alcohol and lipids is that the alcohol is going to dissipate or dissolve that outer membrane, and it washes that crystal violet iodine complex out. So at the end of the alcohol wash, it looks as though gram-negatives don't have any stain to them because it's all been washed away, and which is the reason we have to come back through with a counter stain of saffron, which is that pinkish red stain. It's actually red, but sometimes it can appear pink. So here we have our gram-positive and gram-negative. So tell me which is which. Who's gram-positive and who's gram-negative? If you said this guy was P for positive, P for purple, you're absolutely right. And then this one, red neg, these are our gram-negs. If we were to look at the shape of them, we'd see that they are both rod-shaped. So other aspects or differences between a gram-positive cell wall and a gram-negative wall, the first bullet we've already talked about. Gram-positives have a less complex basal body attachment for the flagella, whereas gram-negatives have a much more complex basal body attachment for the flagella. And let's just look at that again just so that we can remember exactly what it is that we're talking about here. So let's find that slide for the flagella. So here was our gram-positive, 2 rings, and gram-negative. So there's 1, 2. One, 2, 3, and 4. So much more complex configuration. And then we also have for gram-positives, they can be disrupted by lysosome, which is an enzyme that you find in your tears and your sweat and your saliva. Gram-negatives have that endotoxin, that lipid A made that endotoxin. Gram-positives, because of their configuration, are more sensitive to penicillin, whereas gram-negatives aren't nearly as sensitive to penicillin. In fact, they're much more sensitive to tetracycline, which is a more powerful antibiotic. And here's just a picture of that gram-positive and gram-negative cell wall. I'm going to give you a moment, an opportunity to kind of think about some of the things that we talked about as far as the differences between the two and the configuration of that LPS layer. So not all bacterial or prokaryotic cells have a cell wall that is made out of peptidoglycan. And we talked about that in Chapter 3. We can have acid-fast staining cell walls. So we use a slightly different procedure to stain those walls. So acid-fast cell walls are very similar to gram-positive cell walls. In fact, they look, if we gram stain them, they would actually gram stain purple, but they're not really gram-positive. They have a mycolic acid or a waxy lipid layer that's bound to peptidoglycan, which makes it an atypical cell wall. Examples of acid-fast staining cells are Mycobacterium and Nocardia. So those Mycobacterium and Nocardia are examples of acid-fast staining cell walls. And remember, where I put a sunshine, that means that it's important. So here is a picture of an acid-fast staining cell wall. Remember, we talked about how we have acid-fast, it's going to be a red color, whereas the non-acid-fast staining cells are going to be a blue color. So we have our acid-fast staining corded growth for Mycobacterium tuberculosis, which is an example of an acid-fast staining cell. And then these big blue cells are non-acid-fast staining. Those are just actual simple squamous epithelial cells that are found in the lung tissue. So they're just human cells in the lung tissue. We also have not only acid-fast staining cell walls that are atypical cell walls, we have mycoplasmas, which don't have the A cell wall. They have sterols in the plasma membrane that allow for it to maintain its integrity. And then that whole domain of archaea are either wall-less or have what we call walls made of pseudomurin, which is not quite N-acetylmumeric acids because it's lacking that and it's lacking these D-amino acids. So for your exam, I would like for you to be able to tell me examples of atypical cell walls, which consisted of 3 different categories. You have the acid-fast staining cell walls, which have those waxy mycolic acids that allow for that process to take place, mycoplasmas that don't have cell walls, and then you have archaea that either don't have cell walls or they have cell walls that are made out of a derivative of the N-acetylmumeric acid that we call pseudomurin. So if the cell wall is damaged, because remember, the cell wall is a very important structure, because if we get rid of the cell wall, there's a chance that we're going to have osmotic lysis, because that's the job of the cell wall. If we have damage to the cell wall, we're probably going to kill the cell. Lysosomes can digest the disaccharide layer in the peptidoglycan layer. We only see them really being effective on gram-positives because lysozyme doesn't have that same effect on the LPS layer of gram-negatives. Penicillin can prevent the peptide bridges that we find in peptidoglycan. And remember, once again, because gram-positives don't have that outer layer protection like gram-negatives, penicillin is a perfect antibiotic to use for them, whereas for gram-negatives, they have that outer layer. So if penicillin is supposed to work against the peptidoglycan layer of the cell wall, since gram-negatives have an outer layer, then we can't really get to it very easily. So that's why penicillins aren't nearly as effective. A protoblast is a wall-less cell. So we talked about archaea being wall-less cells. Spheroblast is a wall-less cell that would stain gram-positive, and both protoblasts and spheroblasts are much more susceptible to osmotic lysis because they don't really have that cell wall, and the whole job of cell wall is to prevent osmotic lysis. L forms are special classes where there's wall-less, but instead of just bursting like a spheroblast or a protoblast would just burst and succumb to osmotic lysis, they actually just swell into irregular shapes first, so they have a little bit more resistance to osmotic lysis or too much water getting into the cell than you'd find for the other forms, the protoblast and the spheroblast. Now on to the plasma membrane. Job of the plasma membrane is to be selectively permeable, and you've probably heard that term before, which means it lets some things in while other things cannot get in. Plasma membrane for prokaryotic cells and eukaryotic cells is almost exactly the same. In fact, everything that's on this list, on this slide right here, is the same between prokaryotes and eukaryotes. They're both made out of this phospholipid bilayer, which means it has a hydrophilic head, a head that likes the water, and the hydrophobic tail, and the tails are tucked into the inside, so that's why it's made out of two layers, a layer on top and a layer on bottom with the tails in the middle. There are peripheral proteins that hang out on either one side, integral proteins that kind of go and are embedded in the plasma membrane or the phospholipid bilayer. We have transmembrane proteins that go all throughout the sides, and that for any of these proteins, that their basic job is to help to determine to communication and also to form channels or pores. And here's a picture of that phospholipid bilayer as well as these various different types of proteins. The fluid mosaic model is basically, it's the same as you've heard in any of your other classes, is that the plasma membrane is not stoic, it's very dynamic, it's always constantly changing with phospholipids moving and rotating and proteins moving to function, and it has about the viscosity of olive oil. So we say that the way the plasma membrane looks, we call it the fluid mosaic model. And because the job of the plasma membrane is that it has selective permeability, means that some things can get through while others cannot. The only difference between a eukaryotic plasma membrane and a prokaryotic plasma membrane is that eukaryotic cells don't have enzymes needed for ATP or energy production on their plasma membrane because eukaryotic cells have this really cool organelle called a mitochondria. So they don't need that. Whereas prokaryotic cells don't have that organelle of a mitochondria. So to make energy, the enzymes for making energy are found on their plasma membrane. In much the same way, if a bacteria is going to be photosynthetic or undergo photosynthesis, the pigments and chromatophores and thylakoids are found on the plasma membrane as well. So these last two bullets are the only differences between a prokaryotic and a eukaryotic cell. So that's where we're going to end things for the day, and we will talk about how to get molecules in and out of the cell, and we'll talk about some of those structures on the inside of prokaryotic cells and have a brief discussion on eukaryotes. Have a great day.